Hello, my name is Mahek Shah, and this is my informative video for PHYSCI 121 on a new treatment for Duchenne muscular dystrophy called SRP-9001. In this course, we have studied many aspects of DMD, including its molecular mechanisms, symptoms, impact on quality of life, treatments, and their approval processes. Therefore, I have decided to expand on the topic of the approval process of new DMD treatments, focusing on SRP 9001, the most recent treatment that has been proposed and requested accelerated approval. DMD is a rare genetic disorder that affects primarily young males. It has an urgent unmet medical need since there has been no treatment developed that can completely reverse the effects of the dystrophin gene mutation. There are many symptoms which begin to manifest around 5 years of age, including muscle weakness and developmental delay. Therefore, around the age of 5, patients with DMD begin a steroid regimen called deflazacort in order to conserve muscle strength. Additionally, there are currently 4 accelerated approval exon-skipping drugs which have received FDA approval to treat the small percentage of patients with DMD harboring amenable mutations in the DMD gene. However, there has been no gene therapy treatment approved that can apply to all DMD patients until one called SRP-9001 was presented to the FDA about a month ago. This graphic outlines what the drug development process looks like, and I will be touching on all of these topics except for post-market monitoring while discussing SRP 9001. So the first step of the process, discovery and development. SRP 9001, also known as delandistrogene moxeparvovec, is Sarepta's new drug that was developed to change the disease trajectory of DMD to a milder version of the disease, such as Becker muscular dystrophy. SRP 9001 uses an adeno-associated virus, an AAV, in order to deliver its new microdystrophin. Dystrophin is the largest gene identified in the human genome and produces the dystrophin protein of size 427 kilodaltons. This entire protein cannot fit inside an AAV, therefore leading to the innovation of the microdystrophin, which contains only the critical domains of dystrophin. This is exemplified by these two figures, which show the differences between wild-type and experimental microdystrophin. Much of the lipid protein portion of the protein has been removed in the microdystrophin, with more central regions, such as the N-terminus and the cysteine-rich domain, being conserved. The vector genome expresses Sarepta's microdystrophin. It contains essential elements to control gene expression, such as AAV2 inverted terminal repeats, chimeric intron, and a synthetic polyadenylation signal. Expression of the microdystrophin protein is under the control of chimeric MHCK7 promoter to restrict expression to skeletal and cardiac muscle. Unlike other shortened forms of dystrophin expressed from the endogenous DMD gene, Sarepta's microdystrophin is expressed from an AAV vector and is thus regulated differently. Biodistribution analyses indicate that the number of vector genomes per nucleus varied widely amongst tissues, with the highest quantities of vector DNA present in the liver, followed by the heart and skeletal muscles. The second stage is preclinical research. Preclinical research is very important because it is conducted to assess the safety, efficacy, and pharmacological properties of new drug candidates before testing them on humans. These studies play a vital role in providing valuable information on drug safety, determining dosage and formulation, demonstrating proof of concept, and meeting regulatory requirements. They are an essential step in the drug development process, helping researchers make informed decisions and increasing the likelihood of success in subsequent clinical trials. Preclinical studies are often conducted in controlled laboratory settings using animal models or isolated cells and tissues. Although they provide valuable insights, it is essential to recognize that results from preclinical studies may not always directly translate to human outcomes. The proof of concept for SRP 9001 was determined by 3 different studies that showed dose-dependent increases in expression of Sarepta's microdystrophin, demonstrated by immunofluorescence and Western blot in target tissues at 12 weeks post-administration of SRP 9001. Stage 3, clinical development. Clinical trials are an integral part of the journey from scientific discovery to the development of safe and effective treatments, and they play a pivotal role in shaping the landscape of modern healthcare. At their core, clinical trials are rigorous scientific investigations that aim to evaluate the safety, efficacy, and potential benefits of new medical interventions, such as drugs, vaccines, medical devices, or treatment protocols. These trials involve the participation of human volunteers who bravely contribute their time, effort, and sometimes their own health to further progress medical knowledge and pave the way for better treatment options. Clinical trials are conducted in multiple phases, each serving a specific purpose in the evaluation process. Phase 1 trials focus on determining the safety profile and optimal dosage of a new intervention. Phase 2 trials expand the investigation to assess initial efficacy and potential side effects. Phase 3 trials involve larger populations and aim to establish the intervention's overall effectiveness and safety compared to existing treatments. Finally, Phase 4 trials, conducted post-approval, continue monitoring the intervention's long-term effects and evaluate its real-world impact. Phase 1 of the SRP-9001 approval process is called Endeavor, and it is being performed in order to determine the safety of SRP-9001. This study began in 2020 and had 46 participants, and it is slated to go until 2025. The participants of Endeavor received a single intravenous infusion of SRP-9001 on day 1 of the study. The primary result being measured is the change in microdystrophin expression after 12 weeks. However, the study still occurs over the span of 5 years in order to measure secondary outcomes, such as vector shedding, measured through urine and stool samples, AAV antibodies present in the participants, and to see if any serious adverse events occur. Another trial, where Phase 1 and Phase 2 are being performed simultaneously in order to determine safety and dosage, is also being conducted. This is an open-label, single-dose gene transfer therapy study evaluating the safety of SRP-9001. There are 4 subjects who were all administered with SRP-9001, and the primary outcome of this study was to measure adverse effects, while secondary outcomes included changes in the participants' 100-meter walk test results over the course of 5 years. The image shows H&E stains taken pre- and post-treatment for each patient, and there is some improvement visible regarding central nucleation. The name of the third phase is Embark, and it began in 2021 and is scheduled to conclude at the end of 2023. This trial has 126 participants and is the first in this process to use placebos. The experimental group will receive SRP-9001 infusion on day 1, and for the placebo group, they will receive a placebo infusion on day 1. The primary outcome being measured is the changes in results of the North Star Ambulatory Assessment over the course of a year. Envision is the name of another Phase 3 trial that is upcoming and that will also be double-blind and placebo-controlled. Phase 4 of clinical trials is dependent on FDA approval. Only after a treatment is FDA approved will a treatment be able to be marketed and monitored for long-term impacts or side effects. Stage 4, FDA review. SRP-9001 is currently under FDA priority review for a biologics license application, a BLA, which allows for a new biologic product to be introduced to the commercial market. The FDA Cellular, Tissue, and Gene Therapies Advisory Committee voted in an 8-6 vote in support of the accelerated approval of SRP-9001, which will be taken into account by the FDA while making their decision on the BLA. The priority decision was supposed to have been made by May 29, 2023, in order to determine the future of the Phase 3 trial Envision, but the decision has been pushed back until June 22, 2023. In conclusion, clinical trials have many involved stages and steps that are necessary to complete before a drug can be approved for mass use. SRP-9001 especially is an innovative drug that could help many children with DMD with their quality of life, muscle function, and ambulatory abilities. Thank you.